Well, good afternoon, everyone, and welcome to the inaugural Research Grand Rounds. And so this is a new initiative that we're trying to do, and it's multi-purpose. One is, thank you very much. I see house staff, residents here, and what we're hoping to do is get our clinicians and our researchers to actually see each other, work together. Obviously, there's that thing called translational research, where our basic scientists could mix with clinicians and come up with projects, inspire us on the clinical side to actually use the novel investigative techniques that are happening here and bring them into the clinical world. There's a lot of that going on. We're going to try to enhance it even more. But also within the basic sciences, we tend to work in silos a little bit, and to have researchers present to each other as well. It tends to enhance communication and even technical advances in each of particular domain. So very appropriately, our very first inaugural speaker will be Dr. Roberto Bolli. We'll be talking about cell therapy for heart failure. But I want to tell you a lot about Dr. Bolli. I've actually known him in the cardiovascular world. He invited me to be a speaker here a couple decades ago, the first time I was ever at Jewish Hospital in University of Louisville. But he's really been a leader. When you look at the track record for investigations and science and funding here at UofL, a lot of it was initiated by Dr. Bolli. And I won't embarrass him by saying how many millions of dollars he's brought into the university. But I can tell you that, the H-index of 132, it's thousands of publications. He graduated from University of Perugia in Italy. And did research fellowships at NHLBI, and his clinical fellowship was actually at Baylor. He joined the faculty at Baylor, where he actually became full professor with tenure, and then became chief of cardiovascular medicine here at University of Louisville. He held that position for 26 years, and really initiated and became the founding director of the Institute for Molecular Cardiology, and is a university distinguished scholar. He's also the Jewish Hospital Distinguished Chair in Cardiology, and he has been at two different institutions developing a leading research program starting from zero. So I could go on, and then you'd have less time. Next time, hopefully all of you who are online, you'll come, because the food is absolutely amazing. And next time we'll get the food out like 1:45, I think, so that the person can have a full hour. But we're not doing that. You got 53 minutes. Thank you so much for agreeing to do this. And we're looking forward to all your comments and your inspiration for research here at Louisville in the past and in the future.
Yeah, thank you very much, Dr. Williams, for inviting me to be the first presenter at this conference. I'm very honored and appreciate the opportunity, and thank you for all your support. So I changed my title a little bit after I told Jason, and I added the subtitle, A Translational Journey, because that's what I like to spend most of this time today, is describing our translational journey from animal preclinical basic research to a clinical trial. So first, I'd like to start with the acknowledgements, lest that we run out of time for them. I would like to acknowledge all the members of the Institute of Molecular Cardiology, which this year is 30 years old. It's incredible. Seems like yesterday when I arrived here and I started building it. But these include over 100 individuals comprising faculty, fellows, students, and staff, who have done an incredible work, and I would like to thank them for their work. I would like to thank our funding sources, almost exclusively the NIH, and more recently Department of Defense, which together have provided us over $150 million of research grants to the university. I cannot mention every collaborator, obviously, because of the numbers, but I would like to acknowledge those who have contributed to the work I will show today. At the basic level, Dr. Lee, Guo, Long, and Vysotinsky, and at the clinical level, Sohel Ikram and Nachika Tapti and Dinesh Kalra, who is collaborating with us in our pig studies and clinical trials. I'd like to express our appreciation. And finally, Liang Teng, Qinghui Wu, and Luo Li, who are doing an incredible work in the animal model lab of the Institute. So I am probably the only person, or maybe one of the very few, who does not have a financial interest in cell therapy. And the reason I show this slide, because you might think I'm an idiot, but I think one of the reasons there has been so much controversy and conflicting results is that almost everyone in this field has a company or some kind of financial interest in the success of a certain therapy. And I may be wrong, but in my opinion, when investigators have a financial interest, they will affect the way they design, conduct, and interpret their data. And I think that has been one of the major problems. What we need is real investigators who are not driven by financial interest. I will spend a few minutes on general concepts in this field. I mean, this is an enormous field, and I really don't have time to review the whole literature and so on. I'd rather focus on what we did here in Louisville. But I'll spend a few minutes on general concepts and then describe our translational journey here at the University of Louisville. So every time a truly new innovative therapy comes into play, we observe this Gardner cycle. So there is initially a peak of inflated expectation, exuberant, irrational exuberance, as the chair of the Fed said many years ago, where everybody expects things to be really amazing. Then this is followed by a trough of disillusionment when the data don't pan out. And then there is a phase of rigorous methodical research that leads eventually to a steady state of productivity. And I believe that in cell therapy, we are at this point, we are at this point of the cycle, initially at the beginning of the millennium, when cell therapy burst into play, there was absolutely irrational, exaggerated expectations, which were inflated as always by the media. And that was followed by some disappointing results, which led to a very negative attitude towards cell therapy for heart failure and heart disease in general. And I believe that now we are rebuilding this field with methodical, rigorous studies. And I think that in a few years, I personally think, that some kind of cell therapy, probably mesenchymal stromal cells, will be part of a routine treatment for heart failure. So I was encouraged when Eugene Gromwald published this editorial a couple of years ago. He has never worked on cell therapy, so he called me up and we discussed basically the content of this editorial. And I was very happy that he essentially adopted all my suggestions. But this was a very influential editorial because it really called for renewed efforts to bring cell therapy to the clinical arena as a treatment, and particularly trying to convince the NIH to spend more money in cell therapy. So a few basic concepts. A stem cell is a cell that's capable of self-renewal and differentiating into one or more tissue type. That's the definition of a stem cell. Self-renewal, differentiation ability. Now, there are two fundamental types of stem cells. There are the pluripotent stem cells, which are either embryonic or induced pluripotent stem cells, which can generate every layer of the embryo, right? Endoderm, mesoderm, and ectoderm. So essentially, they can produce any cell type in the body. And the adult multipotent stem cells, which have a more restricted differentiation ability that can differentiate into only one or two layers. Perhaps the best example are the mesenchymal stem cells, which are the topic of our research, by the way, which can differentiate into any mesodermal cell. That means adipose tissue, bone, cartilage, fibrous tissue, and other mesodermal tissues. So, as I said, there are two kinds of pluripotent cells, the embryonic stem cells and the induced pluripotent stem cells. You remember Yamanaka got the Nobel Prize for these cells about 12 years ago, maybe 14 years ago. Let me talk about just a couple of minutes about the embryonic stem cells, because these cells have been touted as being a miraculous cure for many diseases, because according to these proponents that can differentiate and regenerate tissues, including the heart. Well, the reality is actually quite different because the media has always got it wrong, and they don't look at the actual data in this field. So there are major problems with using either ESCs or ESC-derived cells in clinical. First of all, there are major ethical issues because they require the destruction of a human embryo. But aside from that, there are a plethora of major medical issues. They produce tumors because of their very undifferentiated state. And of course, the last thing you want to do in a patient with heart failure is to give them a teratoma in their heart. They belong to a different organism and therefore are rejected and require lifelong immunosuppression, which in itself is a disease. They have been touted as being able to regenerate. Well, if you look at the actual data published so far, and by the way, there is not one single clinical trial, controlled clinical trial of embryonic stem cells in heart disease, not one. So all we have is monkey data or some kind of animal model. If you look at those data, the long-term survival of the transplanted cells is very poor. They don't last more than 1 or 2 months, and certainly there are no data that they last more than 3 months. This was a paper that was published when I was editor of circulation research a few years ago from China. They used monkeys, transplanted human ESCs into these monkeys, and you can see by 4 months, there are none left in the heart. They also produce arrhythmias, which is not nice in patients with heart failure. And also, now that we have iPSCs, induced pluripotent stem cells, which essentially have the same characteristic, it's not clear why, what is the advantage of using embryonic cells. So I personally think these cells, the embryonic stem cells and the derivatives, will be abandoned and will not become part of a treatment for heart disease. Now, as far as the induced pluripotent stem cells, it's a little different. They don't pose the ethical issues of embryonic cells, but they do have some major financial problems. Growing iPSCs from a patient takes months and is very, very expensive. Can you imagine doing a 200-patient trial where you have to grow cells from 200 patients? It is not financially conceivable. Furthermore, once you differentiate them, so you could do allogenic, right? You can take a donor and grow cells and give them to a bunch of patients, but in that case, you're running into the problem of rejection again, like embryonic cells. And furthermore, they also have this issue of tumorigenicity because they are very primitive and undifferentiated. So the bottom line is that I believe that the most likely cells to become a therapy for heart disease in the next few years, in the foreseeable future, are the adult stem or progenitor cells, for which we have hundreds of clinical trials and thousands of data, totally safe, by the way. There has not been one single instance of adverse effects related to transplantation of the cells in themselves. There have been adverse effects related to perforation of the heart or some kind due to the delivery of cells, but not to the cells themselves. And I think these are the most likely cells to become treated. Now, we meaning here in Louisville, were part of this NIH network called the Cardiovascular Cell Therapy Research Network, and we were the principal center in 2 major trials. One is the CONCERT-HF trial, which was published in the European Journal of Heart Failure a few years ago. It was a phase 2 study, randomized, double-blind, placebo-controlled, multi-center trial of this NIH network that investigated the feasibility, safety, and efficacy of autologous. And the reason autologous, because now we're using allogenic cells. Autologous have been abandoned, basically. But the reason autologous, this was designed in 2011. That's how long it took to do this trial. So autologous bone marrow, mesenchymal stromal cells, and cardiac CK-positive cells in patients with heart failure caused by ischemic cardiomyopathy. The oversight was enormous. The NHABI, all the 7 IRBs on the network, the data safety monitoring board, and of course the FDA. And this was arguably one of the most rigorous trials of cell therapy to date. You have no idea the amount of oversight that we had in conducting this study. 7 centers, 2 cores, a biorepository, a GMC for the manufacturing facility in Miami, the data collection center that was the NHABI. So very briefly, this trial included 125 patients with heart failure caused by ischemic cardiomyopathy. Baseline was 29%. They were randomized 1 to 4 groups, either the combination of MSCs plus CPC, which are the CP positive cells from the heart, or MSCs alone, or CPCs alone, or placebo. So 4 groups. And the cells were given by transendocardial injection using the model mapping system. That's an example of the electromechanical map that you get when you do one of these NOVA injections. The red is dead, the purple and blue is alive, and the yellow is a mixture of alive and dead. And you inject in the mixture where there is partly dead and partly alive tissue. So as far as the results, there was no significant effect of cells on ejection fraction, LV volumes, scar size, maximal oxygen consumption on the treadmill, 6-minute walking distance, and the NT-proBNP levels. However, the heart failure major adverse cardiac events for MACE, which included hospitalization for heart failure, cardiac death, and exacerbation of heart failure, they showed a major reduction with the c-KIT positive cells. Here you see that the white bar are the placebo patients. In the green bar are the c-KIT cells. There is a 70% reduction. This is at 12 months, 70% reduction in MACE in this group. And also in the group that received the CPCs plus the MSCs, which is the yellow group, there was a similar reduction, 70%. And if you look at the components of MACE, the reduction was driven primarily by a reduction in the hospitalization for heart failure. Again, CPCs alone or CPCs plus MSCs. Looking at the Cox regression analysis of these patients, the hazard ratio using CPCs alone at over a year of follow-up was 0.2. That means an 80% reduction in MACE. With the combination therapy was 0.26, similar reduction, whereas MSCs alone in green had no effect. So, in addition to the MACE, there was also an effect of MSCs this time, not CPCs, MSCs on the quality of life measured by the Minnesota Living with Heart Failure Questionnaire, both at 6 months and at 12 months after a single injection of cells, where there was a significant improvement in quality of life with MSCs plus CPCs. So the CONCERT trial suggests that in patients with chronic ischemic heart failure, now remember, these patients are on maximally guideline-driven therapy, so they're getting the best treatment you can get. A single, just one shot, a single administration of autologous CPCs or MSCs has measurable beneficial effects over the ensuing clinical months in clinical outcomes, namely a reduction in hospitalization for heart failure with CPCs, and an improvement in quality of life with MSCs. The best results were obtained by combining the 2 cell types, which reduce the MACE and also improve the quality of life. And interestingly, neither cell type improved either LV function or reduced scar size. So these results are actually consistent with other major trials of cell therapy in heart failure. The XL DCM trial published in Lancet about 8 years ago also showed a reduction in MACE with cell therapy. And the phase 3 DREAM-HF trial by Emerson Perrin at the Texas Heart Institute showed a significant reduction in MACE at 2.5 years follow-up after treatment. So this is not a kind of a unique result, but it does show that even though we don't have an improvement in cardiac function or volumes, we see an improvement in clinical outcome. And the question obviously is, how does that happen? The prevailing hypothesis, so the short answer is that we don't know. But the prevailing hypothesis is that these cells, the mesenchymal cells, are known to have potent immunomodulatory actions. They are anti-inflammatory, they suppress pro-inflammatory cells and cytokines. And since heart failure is an inflammatory disease, the main idea nowadays is that they work by suppressing systemic inflammation and therefore improving the clinical outcome in these patients. The other study that the CCTRN did was the Seneca study. This was a study published in JACC Oncology a few years ago. In patients with anthracycline-induced cardiomyopathy who develop heart failure. As you know, these patients have a very poor prognosis, and there is really no good treatment for them. This was a phase 1 study, not a phase 2. Actually, this is the first ever study of cell therapy in this patient population. And these patients were on average 18 years after cancer diagnosis, and the F was 33%. They were mostly females. And the main diagnoses were breast cancer and non-Hodgkin's lymphomas. So we had a number of endpoints similar to concepts. 10 of the 13 measures numerically, but not statistically, favor the allogeneic mesenchymal stem cell group. There was a borderline improvement in the 6-minute walking distance and in the quality of life, despite notice very small sample sizes, 11, 12 patients, 15 patients. So this is clearly not an efficacy trial. This was a safety and feasibility trial, with a phase 1 trial. But nevertheless, it did show this signal for efficacy and provided a rationale for larger phase 2 follow-up studies in this patient population, which, as I said, is a very poor prognosis. We are now participating with Josh Sherry in Miami in the DCM2 trial. It's a phase 2 study where we are using allogeneic. I told you nowadays every cell therapy uses allogeneic mesenchymal stem cells in non-ischemic dilated cardiomyopathy. All right, so let's now go to our translational journey. More than a decade ago, we began our efforts to translate 2 ideas into clinical cell-based therapies for heart failure. So I am a translational scientist. I don't like to do basic research for the sake of improvement. It's important, obviously, but what I really want is to bring a basic idea to the clinic and ideally find a new therapy for heart failure. That is my goal. So we started testing 2 ideas. One is intravenous cell delivery. Until now, every trial I showed you used transendocardial injection of cells. And the second one is using repeated cell doses. Until now, every trial I showed you has used a single dose. And the reason they've used a single dose is because you cannot really, practically speaking, do a lot of transendocardial injections. It's too invasive, it carries risk, and patients will not agree. What is the rationale for giving intravenous? Because initially you think, wait a minute, you give intravenous, you're giving cells to the lung. They don't get to the heart, which they don't. That's true, they don't get to the heart. Well, we have learned, I don't have time to go into this, but as we know, one of the major advances that no matter what cell you give into the heart, they do not engraft. After a few weeks, they're gone, they disappear. So we should stop using the R word, regeneration, and instead call this reparative. We are repairing the heart, but not really regenerating new myocytes. The progressive deterioration of left ventricular function in heart failure is caused at least in part by a persistent systemic is the key word, inflammation resulting from activation of the immune system and causing infiltration of the heart by a whole number of very angry inflammatory players such as monocytes, macrophages, T cells, and so on. Now, the spleen is very important in this inflammatory response. We know there are immune cells going from the spleen to the heart, and there is a trafficking of immune cells. It's the so-called cardiosplenic axis. We also know from studies outside of cardiology, mostly in bone marrow transplants or other autoimmune diseases, ulcerative colitis and Crohn's disease, and on and on and on, that intravenous infusion of mesenchymal stromal cells exerts systemic anti-inflammatory and immunomodulatory actions. So we are interested in seeing whether these systemic actions will be beneficial in heart failure, which also has a systemic inflammatory component. What is the rationale for giving repeated cells? Well, that's really, let's say it's a no-brainer, right? When we have a patient with pneumonia, we don't give 1 shot of antibiotic, right? That will not work very well. We treat it with repeated infusions for several days. And so for almost every other disease we are treating, we don't ever use just a single dose of cells. So why should we do that in heart failure? There is actually no treatment I know of for heart failure that works if you only give it once. right? Unless you're putting a VAD or a transplant, and that's one thing that won't work. So we already told you the cells will not engraft because they disappear rapidly. It makes sense to reason that repeated doses may enhance therapeutic actions. We actually did a lot of work in the last 15 years in the animal lab. We tested this idea. This was a rat study where we produced a large myocardial infarction, which was a coronary occlusion in the rat, and then we followed up these animals, and we gave them 3 treatments at 35 days. Let's just focus on the mesenchymal stomach. There are 3 cell types, but let's just focus on MSCs and control, which is basically the buffer. So they were given 3 treatments intravenously every 35 days, and then we looked at what happened to cardiac function. Well, after the first treatment, you see there is a trend for MSCs in green to improve ejection fraction compared to placebo and also the other cells, but was not significant. However, after the 3rd treatment, now everything is significant. So you can see a cumulative effect, right, of giving repeated doses of cells in this model of chronic heart failure. This is the same data looked at in another way. That's the improvement in ejection fraction after the 1st, 2nd, and 3rd injection. See, every time you get a greater effect. Okay, so we went from rats to pigs, which is what the FDA usually requires in order to give us an IND for a clinical trial. So we use Göttingen pigs. Again, same thing. We produce a large infarction, waited 1 month to let the infarct heal. So now you have chronic ischemic cardiomyopathy. And then we gave 3 doses of either placebo, PBS, or 1 dose of MSCs and 2 doses of placebo, or 3 doses of cells at 35 days. Okay, so this is an example of what the heart looks like at the end of the study. You see this large infarction in white after tetrazoline staining. There was no difference in either scar size or variable myocardium among the 3 groups. So we did not really regenerate anything. We did a lot of echoes, baseline, before the first dose, after the first, before the second, before the third, and at the end of the study to analyze every function. And here are the data of this very long study. It took us about 5 years to finish this study. And you can see that at the end of the study, after the 3rd treatment, both a single dose and 3 doses were better than placebo. There was improvement in ejection fraction. However, the delta, the change was greater, a little greater, although not significant, with 3 doses than with 1 dose. Now, we also did MRI, and this is an example of very large apical infarct we saw in these pigs. You can see the 2 sections. And that's how we measure the left ventricular function in these animals. And Dr. Carver has helped us also analyze and interpret this data. So an example of the 3 groups. So in the vehicle group, we start out before, this is before treatment. So that's 30 days after infarction, but before the first treatment, EF was down to 36. Normally, it's about 55 or 60. So it was a drop in EF. And at the end of the study, it's actually even worse. In the pig that received 3 doses, remember, these are intravenous, so the cells are not going into the heart. They're staying in the body. After 3 repeated intravenous doses of mesenchymal stomach cells, the ejection fraction, you can see, improves from 39 to 52. Well, you may say you're just showing us the best example. Probably I am. But these are the data these are the data that, oops. Okay. So the data for all pigs. So before treatment, the ejection fraction was similarly reduced in all 3 groups. So, by the way, this was blinded. This study was blinded. That's another thing. I don't believe any animal study that's not blinded personally, because there is tremendous amount of bias. So we were blinded as to which pig got what, and the investigators doing the analysis were blind. So at pre-treatment, meaning 30 days after infarction, before the first dose of cells, there was no difference in ejection fraction, about 40%. And at the end of the study, 3 months later, there was a significant improvement only with 3 doses, not with a single dose. And this is the delta, which shows the same thing, improvement with 3 doses, but not with a single dose. We also looked by MRI at the left ventricular strain, and there was the same thing, looking at radial strain, similar at baseline, or not really baseline, I guess before treatment. It's baseline is before, in fact, this is before treatment. But there was a significant improvement at the end of the study with 3 doses of cells. So we were not happy with just echo and MRI. We also went in with pressure volume catheters to measure volumes using hemodynamic studies, which is actually the gold standard for measuring ventricular function. Very few people do that, very few. And almost nobody actually does all 3 things together. They either use echo or they use hemodynamic, but they don't do echo, MRI, and hemodynamic. So this was a very comprehensive study. Using the pressure volume catheter, there was again an improvement in ejection fraction, only in pigs receiving 3 doses, and this is the delta. We also looked at the most accurate way to measure contractility, which is the end systolic elastance, that is the slope of the end systolic pressure volume curve. Which is load independent, unlike ejection fraction, and is considered the best assessment of intrinsic myocardial contractility, although not nowadays almost nobody does it anymore. It's only a few dinosaurs like me are still using these things, but if you ask young people about elastance, they look at you with wide open eyes. Okay, so in the vehicle pig, you see that pre-treatment is blue and post-treatment is red. So the steeper the slope, the better ventricular function. So you see at the end of the study, red is less steep than blue, meaning function is deteriorated. In the repeated doses pig, it's the opposite. So the end of the study slope in red is steeper than the before study, meaning elastance is improved. Again, you're going to say you show us the best example, but here are the group data for all pigs. At the end of the study, elastance was significantly improved in 3 doses pigs versus placebo, but not in a single dose group versus placebo. These are the same data. So then we looked at the heart to try to understand how this happens. We know there is no regeneration. And one thing that's interesting using people series, we found a decrease in interstitial collagen in the non-ischemic zone. Now that's the important zone, right? When you have an infarct, the contractility of the ventricle depends on the non-ischemic surviving myocardium. So there was a decrease in interstitial fibrous tissue, which obviously would improve contractility. We also look at hypertrophy, which we know happens in these pigs, also in humans with ischemic cardiomyopathy. And again, in the non-ischemic zone, that's what's really important. There was a decrease in the cross-sectional area of cardiomyocytes, indicating a decrease in hypertrophy in these hearts. We look for evidence of inflammation. Now, the problem in pigs, there are not many antibodies to really sort out all the immune cell types. So we look at CD68 positive cells, which are macrophages, which are the primary culprits, considered culprits in chronic inflammation in heart failure. And there was actually an increase both in the border zone and the non-ischemic zone of these macrophages. So that's a little puzzling until I spoke with some immunologists, because I'm not an immunologist. And it turns out these macrophages can go either way, can be pro or anti-inflammatory. And so our working hypothesis is that by giving these cells, we are promoting a subpopulation of anti-inflammatory reparative macrophages in the heart, which would explain the CD68. We still have to work out more on the nature of these cells. And as I said, it's very difficult because of the limited amount of antibodies in the pig. So in conclusion, in a porcine model of chronic ischemic cardiomyopathy, intravenous infusion of either 1 or 3 doses of allogeneic bone marrow-derived mesenchymal stromal cells improves left ventricular systolic function assessed by 3 independent methods, MRI, echo, and hemodynamics. The salubrious effects of MSCs are more pronounced after 3 doses than after 1 dose. So there is a cumulative effect of repeated doses similar to what we had found in rats. 3 doses of MSCs, but not 1, resulted in a decrease in myocardial fibrosis and hypertrophy and increased microfilm infiltration in the non-infractive zone. So taken together, the results in rats and pigs demonstrate that intravenous infusion of MSCs are beneficial and have a cumulative effect. That's the take-home message of this very long, and also, by the way, very expensive study in pigs. So these results provide a rationale for testing the efficacy of repeated intravenous infusions of MSCs in patients with heart failure secondary to ischemic cardiomyopathy, which is precisely what we are doing with the CATO trial. CATO is the translation of this basic work to the clinical arena. I told you our goal is to bring therapies to the clinic to find the treatments for heart failure. So CATO is the culmination of a 10-year-long, actually more than 10 years, translational journey from preclinical studies in rodents and pigs to clinical testing. There are many more studies, by the way, which I didn't have time to show you that have led to this. I only show you two, but in several. This is a genuine example, as good as you can find, of translational research, which we are blessed to be able to do, because in my experience, when we start with an idea in the basic lab, 9 times out of 10, it does not work. Right? Even though it's a good idea, it's rational, it's plausible, it just does not work. So to be able to actually take an idea from the basic lab to the clinic is a blessing. So the purpose of CATO is to test in humans 2 strategies that we found to be effective in experimental models, already mentioned to you: intravenous cell delivery and repeated cell dose. So CATO is a phase 2A randomized double-blind placebo-controlled multi-center study of a single or repeated intravenous administration of umbilical cord-derived, not bone marrow, umbilical cord-derived mesenchymal stomach cells in patients with heart failure caused by ischemic cardiomyopathy. So the patient population would be what the pig model was intended to represent. This is funded by an investigator-initiated competitive peer-reviewed 4-year research grant for $8 million from the Department of Defense to the University of Louisville. It took me, by the way, several years to get this grant. I won't go into the horrors of applying and reapplying, but finally, thanks God, we were able to succeed. The oversight is unbelievable. We have spent 9 months just to go through all the regulatory and administrative paperwork. The Department of Defense is very heavy on regulations. There's this thing called ORO, Office of Human Research Oversight. There is a central IRB, but there are also local IRBs. There is a Data Safety Monitoring Board, and of course, there is the Food and Drug Administration. So we spent really months and months just to go through all of this, but thanks to God, we finally went through that, and now we are enrolling. So the trial started in July, we started enrolling in March. And now we are enrolling at a crisp rate, and this is the NCP number for the trial. So the organizational structure is a steering committee composed of the principal investigators, the Data Safety Monitoring Board. UofL is the Clinical Coordinating Center. The Data Coordinating Center is this organization called the Clinical in Maryland. There are 3 clinical centers. So we are one of them in Louisville, Miami with Dr. Hare, and Texas Heart Institute with Dr. Perry. And there are 2 cores, the cell manufacturing core in Miami with Dr. Khan, and the MRI core at Hopkins with Joao Lim. The primary objective is to assess the efficacy. This is an efficacy study of either 4 repeated intravenous infusions or a single intravenous infusion of these UC umbilical cord MSCs relative to placebo. And secondly, to assess the efficacy of 4 doses versus a single dose in patients with ischemic cardiomyopathy. So we want to see, first of all, whether intravenous infusion works, either a single or repeated doses. Secondly, whether, if it does work, 4 doses are better than 1. Secondary objectives are to assess the safety and feasibility, which I think is kind of already very likely to be the case, probably not very important. We already know they're safe. And then to examine the effect of intravenous infusion of these cells on the immune system to try to understand how they work. And so we are doing a whole battery of analysis by flow cytometry of circulating immune cells and cytokines and so on. So this is the protocol. It's a busy slide, but very quickly, these are patients with ischemic cardiomyopathy, F less than 40%. We do baseline testing, and then we randomize them to 3 groups. Control group receives 4 doses of placebo at 2 months intervals.
The single dose gets 1 dose of cells and 3 doses of placebo, and the repeated doses group receives 4 doses of cells all at the same time. At the end of this, we do the same studies we did at baseline with assessment of showing the second what we measure at 12 months after the first dose. So the primary endpoint is ejection fraction. Secondary endpoint measured by MRI, by the way. The secondary endpoints are LV function structure, so this is cut size and systolic and diastolic volume and so on, several parameters by MRI. Maximal oxygen consumption with treadmill, exercise tolerance, New York Heart Association class, the quality of life measured by the Kansas City questionnaire, incidence of MACE, BNP, and circulating cytokines and immune cells. So we are going to do 3 comparisons, and I work with the statisticians to make sure we have enough power. We compare 4 doses of cells versus placebo, a single dose of cells versus placebo, and 4 doses of cells versus a single dose. The question is, are cells better than nothing? And if so, are 4 doses better than 1? So this is to give an idea how complicated this study is. These are the laboratory tests and so on. So this is a picture of mesenchymal stromal cells, which are defined by their ability to adhere to glass in vitro and to express markers like CD105, CD90, and CD73. Why did we use umbilical cord cells instead of the bone marrow, which have been used almost in every previous trial? Well, we actually compared human umbilical cord-derived cells with human bone marrow-derived cells, and we found that the proliferative capacity of the umbilical cord cells is far greater than that of the bone marrow cells. These cells here, for example, doubling time is 34 hours, right, for the umbilical cord, and 56 for the bone marrow, and then Ki67 positivity is much higher. And on the reverse side of this equation, cells senescence is also less with the umbilical cord. It makes sense because the umbilical cord obviously is a very young, vigorous, healthy tissue, whereas when you're using bone marrow, you have an adult who may have been eating, and Dr. Williams will tell you this, the wrong diet for several years, which has adverse effects on the proliferative ability and health of every cell, including the stem cells. So the nuclear size is smaller, the beta-galactase activity is less, and the P21 positivity, which is an index of senescence, is also less in these cells. So they are younger. In fact, if you put them on a plate and you count how many there are after 8 days, they grow 10-fold for the adult bone marrow and 60-fold for the umbilical cord. So these cells grow like weeds, literally. And that's the reason we use them. We also looked using the gene ontology enrichment analysis. We did a RNA sequence analysis of the entire transcriptome to look at what kind of genes are being expressed in umbilical cord cells versus bone marrow cells. And as you can see, the genes regulating inflammation are much more expressed in the umbilical cord than in the bone marrow cells, suggesting that these umbilical cord cells may have more potent immunomodulatory properties compared to the bone marrow. So KETO is a novel trial in several respects. It is testing a new cell product, the umbilical cord cells, and 2 novel strategies that may change the field of cell therapy: intravenous administration and repeated cell doses. It is the first study to use umbilical cord mesenchymal stromal cells in heart failure in the United States. First phase 2 study to deliver a cell product by the intravenous route in heart failure in the United States. First study to examine repeated administrations of a cell product in patients with heart failure. As I said, until now, every study has only used a single dose. It's actually amazing. We got some signal for efficacy after a single dose of cells, right? That's in itself, I think, is remarkable. But this is the first time that we are repeating administration. The reason it's the first time because, as I told you, until now, every study used the invasive transendocardial approach. The KETO trial has important implication because compared to with the invasive technique currently used, which is transendocardial injection, intravenous delivery is obviously simpler, less expensive, safer, and can be performed almost everywhere in almost all patients with heart failure, even as an outpatient procedure. This can be done literally everywhere. You can do it in rural areas, in community hospitals that are not specialized to do transendocardial injections. Perhaps even more important, intravenous injection is exquisitely suitable for repeated infusions. You can envision a situation where a patient with heart failure goes to get his intravenous shot of stem cells and then repeats that every 6 months or so. Therefore, intravenous delivery could dramatically expand the impact and application of cell therapy for heart failure. If Plato finds that repeated intravenous infusions of these cells are beneficial in patients with heart failure, this would be a true paradigm shift and a game changer in cell therapy. It really would change the whole field because it would make it so much easier, simpler, safer, and affordable. By the way, affordable even for patients who don't have insurance. This is much cheaper than having to be admitted to the hospital to do any basic procedure. So we would make it available to everybody. We are also doing now, I don't have to go time to get into that because we are at the end of our time, but we are doing a very exciting study of heart failure with preserved ejection fraction. So all the data I showed you so far are HFrEF, right? Heart failure with reduced ejection fraction. Now, HFpEF is just as serious a problem, if not more serious than HFrEF. We have a lot of drugs for HFrEF, but we have almost nothing for HFpEF. And the prevalence of HFpEF, particularly driven by obesity and metabolic syndrome, is increasing. So we are developing a pig model of HFpEF, which is excellent. We feed these pigs what people eat in this country, right? So fat, sugar, salt, McDonald's kind of stuff. And we make them hypertensive. And they're developing HFpEF. And this would be a great model to test new therapies for HFpEF, which are badly needed. So we are very excited about this study. And I'd like to thank you for your attention. And again, thank you, Dr. Williams, for inviting me to give this talk.
So of course I have a nutrition question, but I'm going to hold that for a minute because you showed a pressure volume. If there's any chance you could go back to that one, it's about 15 slides ago. So obviously, for those of us from the 80s, we loved them. We lived by them. It was how we had insights into hemodynamics. And what you showed was the pressure volume loops and the change in improvement in inotropic state that by looking at intrinsic contractility, there you go.
Yeah.
And looking at the slope of the end-systolic pressure volume relationship, which just people don't talk about that anymore, but it was so important back then as we were learning how to deal with this stuff. Now, the other thing that it shows that I was fascinated by is that when you look at repeated dose, there's a subtle downward shift on the diastolic curve.
Right. Yeah.
Very subtle. And I wonder if you analyze that because, there's two things that you'd like to see. You said that the mass of the cells decreased slightly. That should give you a decrease in myocardial mass, which should improve your peak negative DPT,
Right.
And it's or DPDT, and improve diastolic compliance. As you increase your volume, the pressure does not go up as much, and it looks like you've got that there.
Yeah, so that's a great point. You're right. I mean, you and I, maybe a couple of other people in the United States know what we're talking about. Nobody else knows what these things are. We are the dinosaurs and we are dying off, right? And so,
were you talking about doing a half press?
yes.
That's this.
Yeah, and we are doing the same thing with these peaks, by the way. We are measuring elastance and diastolic. So the slope on the diastolic, this is the diastolic pressure volume slope. That is a measure of the stiffness of the ventricle, right, in diastole. So as Dr. Williams pointed out, in this peak, there was a slight decrease in the diastolic slope versus systolic. So we analyzed that. There is a trend, but it's not P less than 0.5. We have 10 pigs per group and
you
maybe
need
like,
a larger-
yeah, we need, and these pigs, each pig is $50,000. So it's really
Right.
expensive.
So they
Really?
can, when of the small sample size, the consistency of the response. So it happened a little bit and it happened to everybody.
Yeah, we can, I think I'll go back and look at that at the consistency. I did not look at that part. But it makes sense, as you said, there is less collagen, right? Less fibrosis, interstitial fibrosis, less hypertrophy. So this slope should also be decreased in these pigs. So I'll go back and look at the data. Thank you for the suggestion, but these are really important. And in general, it's very sad physiology is disappearing, right? Because it's not sexy. So nowadays, if you want to get a grant, you need to say, oh, this long non-coding RNA 375B that activates 77 genes, and we are going to do single cell RNA sequence analysis. Everybody gets excited and they go crazy, and then nothing comes out of it, right? So anyway, so it's good that we're still doing this.
So we'll open up for questions, and I'll ask my nutrition question in a bit.
My question was about sort of when you were, I thought it was so interesting how you were talking about how the inflammatory component within the myocardium is changing. I was curious if you ended up finding identifying, or if you're thinking ahead about different phenotypes where you're seeing in heart failure patients where you're seeing more of that inflammatory component compared to others. I just thought that was so interesting, and if eventually we might see this therapy catered for one phenotype versus another within these reduced patients.
Yeah, it's a great question. So unfortunately, I'm not an immunologist. I wish I was, but immunology, it would take me a year literally to become an immunologist. I mean, it's so complicated. And there are many, what I learned, there are many subtypes of macrophages, as you said, and what makes it complicated, they can shift from one phenotype to another, right? And so they are pro-inflammatory, anti-inflammatory, and even there are subtypes and so on. We cannot sort that out in this pig model because of lack of antibodies. We are hoping to be able to do some kind of RNA analysis in the new study we're doing in HFpEF pigs to try to figure out at the RNA level what kind of macrophages we have. But in this pig study, unfortunately, we cannot. In humans, of course, that's even more difficult because the only way would be to take a biopsy, but that would be difficult. But it's a great question. Yes.
I have a question about it. So great presentation. Again, going back to trying to understand the mechanisms through which these cell therapy may exert a salubrious effect, like you said. Inflammation, reduction in fibrosis, improvement of hypertrophy, systolic and diastolic improvement. What about coronary microvascular flow? I mean, these are ischemic people, and we have to suspect that endothelial dysfunction, and maybe reversing some of that would help.
Yeah, it's a great point, Dinesh. Yeah, that could be another thing. So, by the way, we looked at angiogenesis. One of the effects of the mesenchymal stromal cells is to promote angiogenesis, formation of new capillaries and arterioles. We did not find at the histological level, at least, an increase in capillary density. But that does not mean that there is not an improvement in microvascular function, as you said. Well, the short answer, we did not test that. And we talked about a few days ago, if we could do that by MRI or by echo, that would be fantastic, because particularly in this HFpEF pig model we are working on now, we know, as you said, that microvascular dysfunction is really important. So that could be certainly an explanation.
Yeah, it was a great talk, and we all follow your excellent work for so many years. I have a question, because the whole idea to use mesenchymal stem cells is somehow to mitigate sterile inflammation in myocardium, that they have some anti-inflammatory effect. And now it's a question, why not to use, people are trying to use a secretor from mesenchymal stem cells. It would be an advantage or not. Second, why not to directly maybe use some drugs to inhibit this inflammation, like inhibit NLRP3 inflammasome, or maybe some cholinergic signaling in myocardium, which actually fuels this sterile inflammation. And maybe last question, because it was very interesting part about this Cato trial. You are using third-party umbilical cord blood
Yes.
cells, yeah? I don't know how it is prepared, this preparation, combined donors and expanded.
Oh, okay.
And we know that, for example, in bone marrow transplant, it depends very much on number of passages of mesenchymal stem cells. No, cord blood mesenchymal stem cells are much better, what you have very nicely demonstrated. But as a product, a medical product, mesenchymal stem cells may vary from passage to passage, depending, combination of donors, difference in histocompatibility, antigens, etc.
Yeah, great questions, Marius. Thank you very much. Marius has been a colleague and a friend now for 2 decades, right, Marius? So let me answer the last question first. The cells we are using in Cato come from a single donor, single woman, who donates the umbilical cord, which otherwise is thrown in the garbage, right? So that cord is used to produce 2 to 3 billion with the B cells. We hope that there will be enough for the entire study, but if not, we'll have a second donor. Anyway, each cell product comes from one woman. Number of passages, I'm pretty sure, I can check with Aisha Khan in Miami, but it's 5 or less. These cells grow like weeds. I mean, you need to spray them with a pesticide to stop them, because they grow. So from one cord, we can get 3 billion cells, right? So 5 passages are enough. So they're very young, which is, your point is very good. The more passages, the older they get, they become senescent and so on. These are very young, vigorous, healthy, excited cells, right? Now, as far as therapy for using drugs, that's also a good idea, but you know better than I, because you're an immunologist, that the immune system is very complex and very redundant. So you block one pathway and there are 5 other pathways that can do the same thing. So we had in heart failure the TNF-alpha antagonists that were precisely trying to use a drug to block inflammation, and that did not pan out. There are probably other studies that have also been neutral. And nobody knows why, but I think the reason is that the immune system is so complicated, you cannot just block one pathway and stop inflammation. The advantage of the mesenchymal cells is that they secrete a plethora, really a panoply of anti-inflammatory cytokines, exosomes, bioactive lipids, microRNAs, long non-coding RNA. These are factories that are really secreting things into the tissue and into the circulation, which inhibits inflammation. So you're not just blocking one pathway, you're blocking multiple pathways at the same time. You know very well in graft versus host disease, for example, you give mesenchymal cells that will attenuate the rejection of the graft by the host, right, by the recipient. And in many other autoimmune diseases, they have been very effective in reducing inflammation. So there is no question they can be very effective. And the first question, the secretome, that's also a very good idea. And there are people doing that. They're taking, so let me back up because I didn't have time to go into it. I told you that cells don't engraft, right? No matter what cell you put in the heart, you look after a month or 2, they're gone. So the question is, how do they work? They work by secreting things like cytokines or microRNAs, long-term RNA, and so on. Well, that's the secretome. So why not use the secretome instead of using the cells? So I think it's a good idea. There are people testing that. There have been studies showing efficacy. I personally think cells are superior to the secretomes for the simple reason that you give an injection. Let's say you give an intravenous infusion of secretome, right? Well, the cytokines and growth factors and RNAs will be gone in a day or 2. Right? They're very rapidly destroyed, metabolized. You give cells, they go to the lung, to the spleen, to the liver, wherever they go. They stay there for a few weeks, maybe even longer. We don't know exactly how long, but for a while. And during those weeks, they will release a secretome. Okay, so now that secretome is going to be released continuously for weeks and weeks rather than just one day. So, but I cannot tell you that cells are better than secretome or vice versa. We simply don't have the data. Yes.
I have 2 questions. One is what you were starting about, but
The second question I'm gonna ask is, what about the therapeutic plasticity? So if you complete the treatment, you follow up with the patients for 6 months. Is there a strategy to see whether the efficacy will last and how much? That's the first question. The second is about secretome packing. So basically, when you are injecting the cells and you're giving them an environment where they should have to have the target endpoints. Why don't you use an ex vivo one for the same and then use that secretome?
So you're asking whether at 6 months we see an effect of cell therapy?
Well, whatever it is the
Yeah.
case, like after the completion. Like post.
Yeah, in the animal model, we saw an effect. So we kept these pigs going for like 5 weeks after the last treatment. And the reason is always financial, because every day it's $20 a day. It would be cheaper to keep them in Motel 6 than to keep them in the, facility where they charge us even more than, but it's $20 a day per pig. So keeping them for months and months is very expensive. But at 1 month after last treatment, we saw this effect, and it was cumulative. Now, in rat studies, which are much cheaper, we went out to a year after treatment, and at a year, we still see the effect continuing, which to me, it's actually amazing. I don't know how to explain that other than reprogramming of the myocardium by the cells, but it's amazing. The effect lasting. Clinical trials, like Concept was done 1 year follow-up, and most of the trials have done 1 year follow-up, and we see a beneficial effect at 1 year. Now, whether we are losing the effect, meaning that if we looked at 3 months or 6 months, it would be even greater, which is plausible, we don't know that. Now, the second question was about.
So, when you're putting IV treatment and you're giving the opportunity to the cells to reach to an environment where they can act, release the secret. That is anticipated to work on in 5 years. But can you use the same cell lines to give an external environment to produce that secret and then use them?
So what you're suggesting, take the cells, put them in some kind of environment that, well, they're being cultured, right? So they are being cultured. But you say putting them in an environment that mimics the host. Maybe there are better ways to grow them with, not just with a buffer, with a medium, but with some collagen, with some endothelial cells, some stromal cells, fibroblasts, and so on. And that could improve their secretome. That's possible. Yeah. I don't think anyone has done that, but that's certainly plausible. It's a good idea. It should be looked at.
And that's why we wanted to do research grand rounds. I don't know if everybody was listening, but the last 10 minutes is a brainstorming thing that will help you, and you're certainly helping us. And that nutrition trial, if this secretome is anti-inflammatory, you gotta test it versus a whole food plant-based diet, which dramatically decreases inflammation and improves the immune system.
Yeah.
And it could be that that 41% decrease in heart failure admissions in the REGARDS trial. Nice, but it was observational. It was not randomized. So we don't know what it really is.
Right.
All we have is a signal.
Yeah. And I think we are hoping these cells do what the vegan diet does, which is to reduce inflammation. It would be actually interesting to compare the two of them, or if they have another effect. Before we go, those of you who see patients, remember, if you see ischemic cardiomyopathy, EF less than 40, please send me a text or an email, because we are looking for patients. Thank you.
I got a quick question. You're using whole blood cell, which is excellent idea compared to bone marrow because they're hard to collect. But it's not going to be autologous transplant. So what about the rejection?
So these cells are immunoprivileged, right, from the umbilical cord.
Yeah.
They're immunoprivileged. So again, I'm not an immunologist, but looking at the literature, and they've been used in many other situations. So there are tons of studies with umbilical cord. They are not rejected by the recipient organism, by the cells. So that's what the literature says, because we have not tested that. But we are measuring, by the way, PRA panels in this patient to see if they develop antibodies against the transplanted cells, but we don't think they will.
Thank you.
Thank you.
So are you okay doing the Cardinal Research Minute? Yeah, okay, all right. So Jason? Jason? Let me do the Cardinal, I'm gonna call it the Cardinal Research Minute. Thank you for doing this.